Hello everyone, let us discuss about the largest virus that affects the human, cytomegalovirus, their general properties, structure, transmission, replication and the pathogenesis and laboratory diagnosis, treatment and their preventive measures. General characteristics of cytomegalovirus. It is a double standard DNA virus which is known as the largest genomic virus that affects the human, and it belongs to the member of herpes virus. The three distinct layers of cytomegalovirus particle would be the first the icosahedral capsid and second tegument layer, outer lipid layer, or the three distant layers of CMV virus particle. So the size of the cytomegalovirus varies between 160 nanometer to 200 nanometer. And next structure of cytomegalovirus, glycoprotein is present at the outer layer of cytomegalovirus and nucleocapsid is covered by the envelope which plays a major role in the virus, in which it helps the virion to attach to the target host cell through the cell receptors. Tegument is a layer which is present between the envelope and the nucleocapsid. So as I said before, the genome of the cytomegalovirus is double standard. Cytomegalovirus is a common virus of newborn which infects around 0.5% to 2.5% around the world. So once cytomegalovirus infected to your body, it will retain the virus for life, which means it will sustain your body throughout the life and it may cause recurrent infection. Okay, and it is the most common viral cause of congenital defects in the sense the baby will be affected during the intrauterine life, which can be identified before or after birth. And cytomegalovirus is a opportunistic pathogen in immunocompromised patients or the person with weakened immune system, so they can be infected very easily. And replication of cytomegalovirus is slower than of other herpes simplex virus. Next, let us see the transmission of cytomegalovirus. It can be transmitted through the bodily fluids like saliva, blood, like that, and vertical transmission, as I said before, a congenital defect like that, which can be transmitted through the mother to baby through their growth cycle. And next, blood transfusion and organ transmission can also be a carrier. And next, replication, attachment and entry. Cytomegalovirus attaches to the host cell via the viral glycoproteins which interact with the cellular receptors, and it enters to the host cell through fusion with the cell membrane. Early gene expression. The genes are expressed for transcription and translation to produce proteins involved in DNA replication process and other essential functions, so that the viral DNA synthesis begins at this stage. And next, replication of viral DNA. Using host cell machinery, the viral genome will be replicated in the stage. And next, late gene expression. The late genes are expressed which encodes for the structural proteins to the formation of new virus particle. And assembly and maturations. Once the newly synthesized viral components are assembled into a mature virions in the host cell nucleus and cytoplasm, and it will tend to release. So next, release. Virions are released from the infected cell either by the means of cell lysis or through the process of which is called as exocytosis. The cell lysis will always damage the host cell, and exocytosis will not do so. Okay. Pathogenesis of cytomegalovirus. First, primary infection. Cytomegalovirus enters the body through mucosal surfaces. The initial infection will be mostly asymptomatic, which means the virus will exist in the body without any symptoms. And in immunocompromised individual, it may be like flu-like symptoms, virus dissemination, and cytomegalovirus spreads to various tissues via the infected white blood cells, especially by means of monocytes and neutrophils, so that the virus can able to establish the latent infection in organs. Response to the virus. The host immune system, especially T cells and nature killer cells, responds to control the viral replication. The cytomegalovirus has evolved various mechanisms to evade immune detection and contributes to persistence and latency, which will infect later. Latency and reactivation. Cytomegalovirus can establish latency in certain cell types where the viral genome persists without active replication. Immunosuppression or other infection can trigger the viral reactivation, which may lead to the recurrent infection. Clinical manifestation of cytomegalovirus in immunocompromised individuals such as transplant recipients and HIV patients. The cytomegalovirus can cause severe diseases affecting various organs, which includes the lungs, livers, and gastrointestinal tract. And congenital infection, if a pregnant woman occurs a primary cytomegalovirus infection, the virus can be transmitted to the fetus, which lead to the congenital cytomegalovirus infection to the baby. And next, let us discuss how cytomegalovirus is diagnosed. And first, viral culture. Cytomegalovirus can be cultured from the clinical specimens such as urine, saliva, blood, or tissue samples. And the procedures involves growing the virus in a specialized cell culture and then observing its characteristic cytopathic effects. So, cytopathic effects, which means the damage to the culture. Nucleic acid amplification test or polymerase chain reaction assays can detect the cytomegalovirus DNA in clinical specimen with high specificity. And next, serological test. Serological assays detect antibodies produced by the immune system in response to the cytomegalovirus infection. And next, antigen detection. Immunofluorescent assay, enzyme immunoassay can detect the cytomegalovirus infection for the search of PP65 antigen in clinical samples like blood or tissue specimen. Histopathology, the examination of the tissue sample, often obtained through the biopsy of the infected organ or tissue, which can reveal the cytomegalovirus inclusion bodies in infected cells. Okay. Preventive measures of cytomegalovirus, we should have a proper hygiene in order to prevent the pathogenic virus, and several educational programs should be conducted to give awareness to the people who don't have it. And blood product safety like blood and saliva or any other, during the blood transfusion and organ transfusion we must have a care on it. And we should avoid close contact with the infected patients. So that's it.